Okay, now we're going to move into three-point mapping. So the purpose of three-point mapping is in part the same as what we covered in the previous part of the lecture. One of the things we want to do is just be able to determine how far apart genes are on the same chromosome, specifically in the category where there's three genes that are present. But the other thing we want to do is also determine the order of genes. So let's say we have three genes, A, B, and C, we'll call them, and so there's multiple possibilities for the order of those three genes. So you could have B in the middle, but you could also have A be the gene that's in the middle, or C be in the middle. And so we want to have an experimental strategy that allows us to determine and distinguish between these three possibilities. So which gene is actually in the middle, and then on top of that also know, this is 10 centimorgans apart and this one is 5 centimorgans apart. So measuring both how far apart they are and then the order. And so how do we do that? So this is where the three-point mapping comes into play. And so there's three criteria that need to be met. So this, again, is simply a test cross that we're doing, where one individual parent must be a heterozygote for all three genes under consideration. And so then we cross this individual to a homozygous recessive individual. The phenotypic class must reflect the genotype of gametes of the parents. So in other words, the heterozygote, the one allele must be dominant to another. So you have to be able to link the particular phenotype of the offspring to the genotype of one of the parents. And then finally, a sufficient number of offspring must be produced for representative samples. So this again just deals with the fact that there's noise, and one of the important things of this cross is we have to be able to determine which categories are most common. So before we talked about the fact that the parental gametes are always the most common. And then in this case, we also need to deal with the fact that the double crossovers are the least common. So you need to have enough offspring that you don't have to worry about noise and stochasticity, and you can really determine each of those categories. And so this is why we typically are going to look at like thousands of offspring crosses. And so let's say we have three genes, so these are Drosophila genes. So again, the plus sign means it's dominant. So this is the dominant allele, and then you have a heterozygote for each of these three genes, and so you also have a recessive allele at all three different genes. So there's the V, the PR, and the BN gene. Okay. So we're going to do a test cross. So kind of as before, we have a heterozygote individual, and we are going to cross it to a homozygote recessive individual. And so again, why do we do that? We do that because the offspring of this cross, we can use the phenotype of the offspring to infer the gamete that was produced in this individual. So we know that this gamete is always the recessive alleles of all three. And so now we can really look at what exactly the gamete was of this individual. But now we're going to do it a little bit more complicated, where one, we don't know what the order is, and two, we don't know what the parental gamete is. And so this top kind of lists all sorts of different situations that could have occurred for this heterozygous individual. So is this heterozygous individual, does the PR gene start in the middle, and then are the parental gametes V plus BM plus PR plus? But you could also have a heterozygote individual that looks like this. So the dominant alleles are all on the same gamete. The parental gamete is all dominant alleles, wild type alleles, and then the other gamete is all recessive alleles. And then in this case, the V gene is in the center. So just keep in mind that there's a large number of possibilities that we're distinguishing between. And so which one of these is correct when we do this test cross, and how we use the offspring in order to determine that. And so there's two really key criteria that we need to use. So one of them is to identify the non-crossover class, the parental gamete class, and then the other one is to identify the double crossover class. And the criteria for this is very straightforward. So the parental gametes, the non-crossover gametes, but this is also, you would refer to as the parental gametes, those are the most common, okay? So the most common gamete, the most common class right here, represent the parental gamete combinations. And then I'll show you in a second how you use the double crossover. So these are the least common, and how we use those in order to infer the order. So, if we add up all of the offspring that were these, we have 467, so 42.1% are this. And so you'll notice that these are all, the gametes are opposite of each other. And then in this case, there's only 7.8% that represent this, and that's how we determine that that was the double crossover. And so now we can use this to determine the parental gamete, so plus VVM. An example of that is this. Another example is of this. And then there's also a third possibility where the V gene is in the middle, but that particular combination isn't present up here. So we just use this information to at least infer the parental combinations. And so now in order to figure out the order, we have to use the fact that what the double crossover is. And so we know that the parental gamete is some sort of combination of plus VBM and then PR plus plus. We just don't know the order. And so this set right here, we consider what would happen in the double crossover in the event where each different gene is in the middle. So in the first case, V is in the middle, and then in the third case, the PR gene is in the middle. And so the double recombinant will start with the orange, and then the first crossover will be in between the PR and the V gene, and then if we continue to follow this back over the other crossover, we can now infer the double recombinant genotype. And so plus BM, so that's just written right here, and then if we follow this situation right here, PRV plus, so that's the second one that would be created. So this is what would happen if V was in the middle. But now if we switch which gene is in the middle and kind of do the same thing, what we end up with is a different type of gamete. So plus V, PRBM plus. Okay, so that is not the same as the situation up here. And then if the PR gene is in the middle and we do this, then we get a different set of combination between these alleles. So the double crossover would produce plus plus plus, the three dominants together and then the three recessive together on a situation. And so when we look at what was actually produced, so if we look at the results of this cross, we find that, the plus 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 was actually the double crossover gamete. And so we knew that because they were the least common gamete among this cross. And so we know that this is the situation that occurs. And so the results of these two categories are simply what's produced when a single crossover occurs. So now that we know that PR is in the middle, we look at what would be produced from, this cross right here. And so that would be VPR plus, and then plus BM, and then the other possible single crossover is when it occurs between these two genes, and then these are the ones that would be produced. So this is kind of the strategy that we use in order to go from all of this uncertainty into the situation where we know exactly what the order is, along with the parental combination was. And so the parts of the activity that you'll do will be to actually calculate you can also use this data in order to calculate the map distance between each of these genes as well. So that is the extent of the lecture portion on Chapter 5. So again, this chapter, it's very important to work through all of the example problems in order to really figure out how to do this. This is the thing where it has to be done. It's just like a puzzle that you just have to get practice at. But once you get practice at it, I think it's pretty straightforward. So that's the rest of this lecture. Thank you.